Chapter 5 Carbohydrate Digestion. Digestion is the great secret to life. What the patient takes beyond his ability to digest does harm. While the underlying causes of various intestinal disorders cannot be stated with certainty, faulty digestion and malabsorption of dietary carbohydrates may, in large part, be responsible for these disorders. Carbohydrates refers to starch and disaccharide starch molecules. Both require digestion before absorption. As we have seen in previous chapters, this can lead to more serious malabsorption of all nutrients due to injury to the intestinal surface. The carbohydrate diet most often corrects malabsorption, allowing nutrients to enter the bloodstream and be made available to the cells of the body, thereby strengthening the immune system's ability to fight. Further debilitation is prevented. Weight can return to normal, and ultimately there is a return to health. Malabsorption is the inability of the cells of the body to obtain nutrients from foods eaten. As a result, the caloric energy, vitamins, and minerals are lost, as all parts of the body are deprived of the proper nourishment. There are many places in the gastrointestinal tract where problems could lead to malabsorption. If food travels too rapidly through the intestinal tract, as happens most when diarrhea is present, there is insufficient time for large food molecules such as starch, fat, and protein to be broken down by various enzymes, and consequently, absorption into the bloodstream is seriously impaired. If a poorly functioning pancreas does not deliver sufficient digestive enzymes to the small intestine to break down large molecules of protein, fat, and starch, absorption of these nutrients will not take place. However, a large number of research reports point to a later step in digestion as the site leading to malabsorption in many intestinal disorders. This last step in the digestive process occurs at the microvilla of the cell membranes of the intestinal absorptive cell. The membranes of the cell lining the intestinal tract serve as more than a passive barrier between the contents of the digestive tract and the bloodstream. When the digestive tract is functioning normally, the membranes of these gatekeeper cells actively participate in the last step of digestion, as well as aiding in the transport of nutrients into the bloodstream. The last step in carbohydrate digestion takes place at the minute projections called microvilla. Only these carbohydrates, which have been properly processed by the enzymes embedded in the microvilli, can cross over the barrier and enter the bloodstream. This is where the milk, sugar, lactose, and sucrose are split apart. This is also the site of the last step of digestion of starch from foods such as grains and potatoes. Figure 7 summarizes the steps involved in carbohydrate digestion in the gastrointestinal tract and lists the microvilli enzymes which carry out the last step of the digestive process. The structure of the intestinal surface is dramatically altered during the intestinal disease, and as a result, digestive activity is seriously inhibited. This makes the last step in the digestion of these carbohydrates difficult, if not impossible. The location of the sugar-splitting enzymes, the disaccharadases, in the membranes of the intestinal cells makes them very vulnerable to damage from many sources. A vitamin deficiency of folic acid, for example, and or of B12, may prevent proper development of the microvilli, which carry the disaccharadases. An abnormally thick layer of mucus produced by the intestinal cells can prevent contact between the microvilli enzymes and the disaccharadases, lactose, sucrose, maltose, and isomaltose. In addition, irritating or toxic substances produced by yeast, bacteria, or parasites, which have invaded the small intestine tract, can cause damage to the intestinal cell membranes, destroying their enzymes. Conditions involving the small intestine that are frequently associated with deficiencies of lactose and other disaccharadases are celiac disease, malnutrition, tropical sprue, cholera, gastroenteritis, infant diarrhea from any cause, pellagra, irritable colon, post-gastrectomy, removal of part of the stomach, soy protein intolerance, intolerance to cow's milk, intractable diarrhea of infancy, parasitic infections of the intestine, cystic fibrosis, and Crohn's disease. In addition, lactase deficiency in ulcerative colitis is well documented, as was noted in Chapter 2. The first enzyme to suffer damage is usually lactase, but often there is a combination of enzyme loss involving sucrase, isomaltase, and less often maltase. The enzyme lactase is depressed earlier than the other disaccharide splitting enzymes in intestinal disturbances such as celiac disease and other conditions where diarrhea is present, and is the last of the microvilli enzymes to return to normal after intestinal disease has subsided. In fact, lactase may be permanently depressed after severe malnutrition and tropical diarrhea, and a deficiency of lactase may be the sole legacy of some previous disorders. It is difficult to prove the absence of disaccharidase activity by present medical techniques. A biopsy sample of the small intestine during intestinal disease may show that enzyme activity of disaccharidase is normal. However, upon feeding lactose, sucrose, and starch, cramping, diarrhea, and vomiting will follow. This apparent contradiction could be due to a lack of contact between the enzymes and sugars caused by the mucus barrier referred to in chapters 2 and 3. When a biopsy sample does indicate that there is a deficiency of disaccharidase enzyme activity, the reason could be a primary genetic problem or a secondary problem caused by a direct injury to the intestinal cell surface with loss of the microvilli and a flattening of the cell itself. Among those factors which lead to injuries of the intestinal surface are malnutrition and irritation caused by substances produced by microbial growth. The sugars then remain undigested in the small intestine. Their presence in the lumen interior space of the intestine causes a reversal of the normal nutritional process. Instead of nutrients flowing from the intestinal space into the bloodstream, water is drawn from the intestinal lumen. The water carrying nutrients is lost in abdominal intestinal function, diarrhea, and the cells of the body are deprived of energy, minerals, and vitamins. Most seriously, the sugars remaining in the intestinal lumen provide energy for further fermentation and growth of intestinal microbes. The increasing levels of irritating substances given off by a growing microbial population cause intestinal cells to defend themselves. Mucus-producing cells, goblet cells, which are normally present in the intestine, secrete their product to cover and protect the naked free surface of the intestinal absorptive cells. The small intestine responds to a disruption of the normal balance by producing more goblet cells, which increases the secretion of intestinal mucus. As the integrity of the small intestine is further threatened by microbial invasion and by the products it produces, a thick mucus barrier forms for self-defense. The enzymes embedded within the absorptive cell membranes cannot do the job for which they are designed, to make contact with the split certain sugars in the diet. If the goblet cells become exhausted and there is a limit to their valiant efforts to defend the absorptive lining against irritation, the naked intestinal surface is subject to further ravaging. It is very possible that at this stage, ulceration of the intestinal surface, as seen in ulcerative colitis, can occur. This might also explain how certain proteins, as gluten, can inappropriately enter the interior of the absorptive cells and destroy them. Sometimes, but not often, even the absorption of single sugars is disturbed because of severe injury to the absorptive cells. But this extreme condition is usually diagnosed by routine hospital tests. Sometimes the invasion of microbes into the small intestine is so pervasive that yeast, for example, will be found in the esophagus. When it is suspected that yeast invasion is widespread, the oral infection, thrush, would be an indicator. It is wise to cut back on honey ingestion at the beginning of the dietary regimen. Amount of honey in recipes should be decreased by at least 75%. The amount of honey may be increased as the condition improves. The indigestibility of starch by even healthy people is only recently receiving attention. Some starchy foods which are assumed to be digested completely are in fact incompletely digested by most people. In those people with intestinal disorders, the digestibility of starch is even further affected. Because the breakdown of starch eventually results in the formation of disaccharides, maltose and isomaltose, most starches must be avoided unless they are specified as permissible in Chapter 10. Some foods in the specific carbohydrate diet contain starches which have been shown to be tolerated. These are the starches of the legume family: dried beans, lentils, and split peas, no chickpeas, soybeans, or bean sprouts. The legumes which are permitted must, however, be soaked for at least 10 to 12 hours prior to cooking, and the water discarded, since they contain other sugars which are indigestible but which can be removed by soaking. The legumes may be introduced in small amounts at about the third month of the diet. The starches in all grains, corn, and potatoes must be strictly avoided. Corn syrup is also excluded since it contains a mixture of short-chain starches. Carbohydrates found in foods. 1. Single sugars, monosaccharides. These sugars require further splitting in order to be transported from the intestine into the bloodstream. They are glucose, fructose, and galactose. Glucose and fructose are found in honey, fruits, and some vegetables. Galactose is found in lactose hydrolyzed milk and in yogurt. 2. Double sugars, disaccharides. These sugars require splitting by intestinal cell enzymes. There are 4 main disaccharides: lactose, sucrose, maltose, and isomaltose. Lactose is found in fluid milk, dried milk powder, commercial yogurt, homemade yogurt, which has not been fermented for 24 hours, processed cheeses, cottage cheese, cream cheese, ice cream, some sour creams, whey, 70% lactose by weight, and many products which have added milk solids or whey. Many drugs and vitamins and mineral supplements have added lactose. Sucrose is table sugar and is found in processed foods such as gelatin, desserts, ketchup, cereals, and many canned foods and some frozen preparations. There is a small amount of sucrose in some pasteurized honey, but it has been shown to be tolerated by those on the specific carbohydrate diet. Unpasteurized honey contains virtually no sucrose, since an enzyme in the honey splits whatever sucrose may be present. Some fruits and nuts contain small amounts of sucrose, but they may be used and have been included in Chapter 10. As fruits ripen, some of the sucrose they may contain is split by enzymes within the fruit. Maltose and isomaltose are found in sources such as corn syrup, malted milk, and candies. However, most of the maltose and isomaltose which is presented to the intestinal cells for digestion comes from dietary starches. Starches are long chains of glucose molecules, which are digested in part by enzymes from the pancreas and saliva, and are left as the disaccharides maltose and isomaltose, to be split by microvilli enzymes of intestinal cells. 3. Starches, polysaccharides. Starches can be of 2 types called amylose and amylopectin. Most vegetables contain both types in various proportions. For example, some kinds of rice contain small amounts of amylopectin starch and large amounts of amylose starch. Other types of rice contain only amylopectin starch. Like rice, some genetic strains of corn contain a very highly branched amylopectin type of starch. Sweet potatoes or yams also only contain amylopectin starch. It appears that genetic breeding, which attempts to change the protein content of certain crops, also affects the type of starch formed by the plant. The varying proportions of different kinds of starch might affect the ability of the intestine to completely digest them, or the proteins of certain plants may prevent the starch from being completely split. It is interesting to note that one group of researchers has found that some intestinal bacteria are made more virulent by the presence of undigested cornstarch in the urine. Vegetables that contain more amylose than amylopectin starch are simpler to digest because the glucose units which make up the starch molecules are arranged in a linear fashion in amylose starch and are readily exposed to digestive enzymes from saliva and the pancreas. The links holding the glucose units in this linear arrays are split until the chains are reduced to only two chemically linked glucose molecules called maltose. By comparison, amylopectin molecules contain glucose units which form branches. When the amylopectin molecules have been partially digested by pancreatic enzymes, the fragments remaining for the last step in digestion by microvilli enzymes are both maltose and isomaltose. Recently, Dr. Gunja Smith, the associate, proposed that the amylopectin starch molecule is even more highly branched than was thought originally. According to Dr. Gunja Smith's diagram, the interior branches appear less exposed than the exterior branches. It is therefore possible that pancreatic digestive enzymes cannot reach the interior links and that parts of the amylopectin starch molecules escape digestion, remain in the intestine, and increase microbial fermentation. At present, very limited information is available as to the amount of amylopectin and amylose starch present in the many kinds of grains and other starchy foods. Fiber. Fruits, vegetables, nuts, and grains contain various components termed fiber, which are indigestible by the enzymes of the digestive tract. Fiber from fruits, nuts, vegetables, including dried legumes, is allowed on a specific carbohydrate diet, but all other forms of grains, including bran, is not permitted.